We have one final presentation to wrap up session 3 on nanomedical structures and devices in development today. And I do think that Dr. Benita, our final presenter, deserves an extra special award for his patience in what has been a very long conference day. Dr. Benita also deserves an award for having traveled to the conference from the furthest distance, traveled here all the way from the Hebrew University in Jerusalem. And Dr. Benita is a recognized expert in the field, in addition to being a fellow of the American Association of Pharmaceutical Scientists. He's also a professor of pharmaceutical sciences at Hebrew University and has co-authored over 153 publications. Please join me with a warm and not belated round of applause for Dr. Simon Benita. Thank you so much for your patience.
No, it's okay. Thank you very much. This is the red one? Okay.
Yeah, the green one is
the green one, yes. Thank you.
Red is the laser.
First of all, I would like to thank you very much and thank the organizing committee for giving me this opportunity to interact with so outstanding scientists in interdisciplinary areas. And of course, to thank very much Ms. Temple and Genesis for their warm hospitality and efficient assistance all over the world coming from a long way here. And for all of you, because it's very late, I will try to show to you a pharmaceutical point of view of a drug delivery system as we are working here. I will today speak about antibodies conjugated to nanocarriers, which contain chemotherapeutic drug for improved cancer therapy. I will skip about polylactate, but one very important issue here will be that these polymers, polyesters, are approved by FDA for intramuscular and intradermal delivery, not for intravenous delivery for the time being. The only nanoparticle approved by FDA in the market is Abraxane, and in fact it's a paclitaxel conjugated to an albumin nanoparticle. Here, what I would like to show is we were dealing with drug targeting. How can we bring a drug, a very cytotoxic drug, to a very specific pathological tissue? We took advantage in cancer of a lot of cells that overexpress antigens that can be specifically recognized by antibodies. We work mainly with 2 antibodies. One of them, I will show you some of our animal data, will be about trastuzumab, that is well known, and also for cancer, of course. In a lot of cancers, mainly for breast cancer, but also for prostate cancer. Okay. Now, these antibodies make a major impact in cancer. There are at least 8 commercial antibodies in cancer, and mainly in hematological malignancies. Now, what we have done and take advantage of these antibodies were to conjugate these antibodies to nanoparticles of polyester here, as you can see. And the main important thing, there are already 1 antibody conjugated to a cytotoxic drug in the market. And of course, it didn't do very well because we had 1 antibody conjugated to 1 molecule. While here, with a nanocarrier, you may have up to 20,000 molecules of an chemotherapeutic drug that can be incorporated in 1 single nanoparticle, whereas this nanoparticle can be conjugated to few tens, hundreds, or even thousands of molecules of antibody, as you can see here. Overall, you can also see that we have pegylated these nanoparticles in order to prevent the rapid elimination by the reticuloendothelial system. Now, what is this advantage of what we are defining as immunonanoparticles, as you can see here? First, we have a very good cell-specific delivery. We can add more than 1 antibody, as I can show you here. We can have a high affinity for a very targeted cell or tissue, and we can incorporate even more than one drug in the same nanoparticle. Now, the objectives of this study were first to evaluate the cytotoxicity, binding, and uptake of these immunonanoparticles in different cancer cells lines, to determine for the first time quantitatively the affinity kinetics parameters of these immunonanoparticles using the BioCore technology, to estimate the pharmacokinetic profile in mice, and finally to assess the efficiency in a well-established tumor-bearing mice model. We prepare these nanoparticles, and one of the most important issues here, this is a well-known method already developed and more than 20 years ago. We here add the linker to these nanoparticles, and the linker was synthesized in our group, as you can see here, by adding a maleimide group to a stereoamine fatty acid to be incorporated in the nanoparticle. Now, this maleimide will react with antibodies that have a thiol group in one of the parts of this antibody, and to prepare this thiolation, we use the TRAT reagent, as you can see here. Finally, you can have from one side the nanoparticle with a very surface active maleimide group that can react with antibodies that have a thiol group, and you will have a covalent here, molecular interaction and conjugation between the antibody and the nanoparticle. The drug that you have tried to incorporate was, in the beginning, paclitaxel. We didn't succeed because paclitaxel has been expelled, and a lot of crystals were detected in the dispersion aqueous system of the nanoparticles. Therefore, we have synthesized a lipophilic derivative of paclitaxel, palmitate, as you can see here. We, of course, characterize this molecule, and we incorporated it efficiently in these nanoparticles that you can see here. And now we have a drug. We have initial drug loading. You can see the observed content. The loading efficiency was 62%. And we could not detect any crystals in the nanoparticulate delivery system. This is one of the most important issues, is to have to identify and determine that you don't have nanocrystals of a drug in presence of the nanoparticles in your system. The first molecule that we have antibody investigated is what H-ferritin recognizing antibody from a company called MAT Biopharma, MB8LK. And this one has been already used in phase 1 and phase 2 study for refractory Hodgkin disease. And we have a monoclonal antibody that we have developed, and we used this antibody called native MB8LK, as you can see here. The normal one has 11 thiol groups, whereas modifying this antibody will provide us more than 30 thiol groups to react with our surface active nanoparticles. I will show you the data here of the nanoparticles, immunoparticles, the size, the zeta potential, the concentration of antibody conjugated to the nanoparticles. We used 1 milligram per ml, and we can find here 0.77 milligram, making us a drug loading of 70% and a conjugation efficiency about 80%. We also checked the binding and uptake of these immunonanoparticles with CAPAN1 cells, which are human pancreas cancer cells. And you can see clearly here that we have a binding of these immunonanoparticles and also an uptake inside the cells. This also was calculated not only by fluorescence, but also by radioactivity, and we also use other methods, including fluorescence fluorometry, and you can see here that these nanoparticles clearly are located, have a very good uptake, and most importantly, we can see here when we have conjugated with FITs that the immunonanoparticles can be localized in the cytoplasm and not in the endosomes, as you can see here, because there is no merging between the green and red color, where we have here a very good orange color, whereas here we can see clearly the red and green. There is no co-localization, and therefore the immunonanoparticles escape from the endosomes. And then you can see here a cytotoxicity of these immunonanoparticles as compared to the native drug and also to the nanoparticles, as you can see here. Finally, we wanted to quantify the binding and how it affects the specific recognition of the antibody. For that, we used the surface plasmon resonance. We did a lot of validations. I will skip because we don't have time here, but you can see from the data, and we did all the experiments, and this will be submitted soon to a paper for publication. You can see here clearly the drug, the antigen, and then we have used the antibody, native antibody, and also the immunonanoparticles. And you can have here the data about the constant of dissociation, and you can see that the antibody, the native antibody, and the immunonanoparticles have more or less the same constant of affinity, clearly showing that our system and its experimental conditions used in this case did not affect the specific activity and the native activity of the antibody. Now we move to the second antibody. Simply, we didn't do biocore because we didn't have with this antibody, trastuzumab Herceptin, the clear and very clean antigen needed for the biocore studies. Therefore, we did it with the IMB8LK because we could have a very good antigen H13, human H13. Now, with trastuzumab, we did also all the thiolation, as you can see here, and you can see clearly that the thiolation is not affecting the specific recognition of the cells, and you have here SKBR cells, and you can see the native antibody and the antibody after modification and reduction with tau reagent. There is no inhibition. In this cartoon, you can see the size, the antibodies, the number, about 63 molecules of antibody per nanoparticle. You can see the concentration of the drug, and also you can see here that they have reacted. Each dark point here is in fact an antibody on the surface of a nanoparticle, and we have a good agreement between the experimental and the calculations that you have seen above. Now, one of the most important things is antibodies cannot be stable over time on storage, and most of them have to be lyophilized. And now we tried, and we collaborate with a group in France to have an idea about the possibility, if you want to design a viable commercial product, what will be the long-term storage of these immunonanoparticles. This is very important. As you can see here, we have the data. After 3 months, you can see here that the immunonanoparticles keep their specific recognition, and most importantly, after 2 years, you can see them stored at 4 degrees. The immunonanoparticles keep all their activity, and they do recognize still the antigen. Now you can see here also that these immunonanoparticles in PC3, which is prostate cancer, refractory cancer, a human refractory cancer, that they can exactly recognize the cells, and you have here a very good uptake and even binding, as you can see here, with radioactivity, and we have a huge binding uptake as compared to the nanoparticles offered to the drug. Now, these have also some efficacy, and you can see here that they can reduce the number, and the survival is markedly reduced. And also, they are not activating the complement. This is very important, as you can see here. You can see, for example, back, yeah. You have here a positive control, 2 donors. We have Taxol, which is the product in the market. The vehicle of Taxol in the market do activate the complement, and you have here the nanoparticles and the immunoparticles, and they are not activating the complement. We move to a pharmacokinetic and biodistribution study, as you can see here. And from this study, we can see here the data of the pharmacokinetics. This is healthy in mice. And I will show you now the data. You can see one very important issue. How behave the antibody when conjugated to the nanoparticles? First of all, you have here the drug by itself. You can see the terminal half-life is 8 hours. In the nanoparticles is 40 hours, and in the immunonanoparticle, 20. Now, what this is very important to know is that the native antibody in beige atemic female mice has an half-life of 75 hours. It's clearly showing that the antibody is losing its intrinsic properties when conjugated to the nanoparticles. In fact, it only is here as a targeting moiety, not acting anymore as an antibody, because it is connected and conjugated and coupled to nanoparticles. Whereas the bioavailability, as you can see here, the absorption and the mean residence time is not different between the nanoparticle and the immunonanoparticle. Now, the biodistribution study here, we had a little bit surprise, and a good surprise. You can see in the liver and the spleen, which are the main organs of a reticuloendothelial system, that nanoparticles are accumulating more than the immunonanoparticles. You have lower figures for the immunonanoparticles than for the nanoparticles. By conjugating an antibody, we added some steric protection to the nanoparticles. This is why we have less accumulation in the reticuloendothelial system. You can, in fact, now can see clearly that we have a more pronounced stealth character of immunonanoparticles over nanoparticles owing to the presence of monoclonal antibodies. And then we move to an orthotopic prostate cancer model in mice. What we have done, we have collaborated with an urologist, as you have seen in the beginning, and we have injected in the prostate of male mice the cells, and we followed exactly the conversion, the proliferation of the cells, and this was a very important PC cells. They were encoded with luciferase, and you have first to know that herceptin by itself has no activity on prostate cancer, resistant prostate cancer. You can see here how the tumor is developing with time. This is only for a control. After a few days, when you inject in the animal, and slowly, slowly, you see the metastasis that spread out of the body of the animal. And we can follow this with a CDD camera by injecting luciferin, and only the cancer cells that are still active will be detected by the camera, as you can see here. And you can see with time that the tumor is growing up, and even you can see the metastasis circulating in the whole body of the animal. Now, we wanted first to see that we have a validated model, and this prostate cancer overexpress HER2 receptor. And you can see it clearly that it did. And we have used 4 different formulations: saline, paclitaxel palmitate solution, the nanoparticles, and the immunonanoparticles. All of them have been injected on the identical dose. And we waited, for example, to have a very clear increase of tumor before the injection. We injected in these mice 3 consecutive treatments of different formulations, as you can see here, and you have the data here. The first injection began after 54 days, when we have a very clear increase in the tumor, and we followed by the camera, and you can see that after 105 days, the smallest volume is elicited by the immunonanoparticles, as you can see here. And this is statistically significant, clearly, but I have to be very frank with you, we could not eradicate the metastasis. We did all the pathology, We reduced markedly the tumor, but the metastases were there. We could not eradicate, and we are asking reasons for this possible reason of incomplete eradication of a tumor and mainly of the metastasis. First, probably we have a problem with the interaction of the immunonanoparticles with the tumor. The first thing is the paclitaxel the most effective drug to treat this kind of cancer? Probably not, because the drug that it is approved by FDA for prostate cancer treatment is docetaxel, which is a different derivative of taxane and not paclitaxel. Second one is probably we have a problem of neovascularization of a tumor. This solid tumor probably increase the neovascularization, prevent from the immunonanoparticles to penetrate within the tumors. Now we are prospecting this kind of data, and we are now trying, and we are repeating this kind of experiment now with two different approaches. One, we are probably going to use two different antibodies, one for targeting, one for anti-angiogenic, or on the contrary, we will still remain with one antibody, and we will add an anti-angiogenic agent within the nanoparticles. One of these two will be probably used, and we are trying to incorporate docetaxel instead of paclitaxel in the nanoparticles. I believe that in the future we will be able to report, but what we can say up to now, that we have identified optimal conditions for the manufacturing of immunonanoparticles comprising significant drug contents. These immunonanoparticles were shown to be specifically binding and to overexpressing antigens recognized by the antibodies that did not lose their activity. They were shown to have drug penetration into the cells. They were more toxic than the nanoparticles to the cells. The affinity was not affected by the conjugation process, and these immunonanoparticles did not activate the complement system in ex vivo assays. Finally, the behavior of the nanoparticles and immunonanoparticle were similar. The coupling of the nanoparticle, of the antibody to the nanoparticle did alter the pharmacokinetic profile of the antibody, and they elicited a very marked anti-tumor effect, although we didn't eradicate the tumor. Thank you for your attention.
Excellent presentation, Dr. Benita, and really provocative, I think.
I hope so.
Yeah,
I am here to hear things and
yeah,
learn from you.
from the
If I
people
hope so,
circling the microphone, it's provocative. First question, please.
Anita Sciotti, Division of Nanomedicine, Houston. I have several questions. It's a great presentation, indeed, a lot of work. Very jealous. But anyways,
Be jealous, you are young.
I have a few questions. The first one is about the fact that you don't use PEG, and there is a great deal of talking about the need of PEG to increase the circulation time, the half-life. Well, you don't have that need, and so I wanted a comment about this, but that is not the only question. The second one is about the zeta potential value of your particles. It's negative, and actually the cells tend to like better positively charged particles. So I wanted a comment also on the internalization rate, and if you have ever tried to switch to a positively charged particle and check if it's more efficient. And I don't remember the third. Maybe I'll remember it.
You can ask me first. Okay, I will answer all your questions because we are aware about this question. First of all, we didn't know in the beginning that nanoparticles without pegylation will have a long circulation time. And this is why we preferred, and we did a lot of studies with nanoparticles with different extent of pegylation, from 10% up to 100%, okay, with a PLA. And we recognize that we don't need so much, and today we use only 16% of pegylation, just to keep it inside, because we never know. The second question is how much, and this is the provocative, and I have a problem because we are planning a clinical trial within 2 years from now, how much of our antibody remains on the immunonanoparticle when they circulate in a human body, not in a small animal. The problem with small animals that a lot of people did succeed in animal experiments and failed in clinical trials. And the reason is if we will lose some of our antibodies. And the question today was in the literature that it is enough to have 12 antibodies on one single nanoparticle to get targeting. We do not believe that it will be sufficient for human clinical trials. We believe that it will be in the number of hundreds and not in the number of 10 or 12. And this is why for the time being we have between 60 to 90, but we increase it to 300 and even more. Now we have used immunonanoparticles negatively and positively charged. We did it because we have also cationic nanoemulsions, and it is true. If you will take a nanoparticle positively charged as compared to a negatively charged, you will have an increased uptake in the cells because of the electrostatic attraction between the negatively charged membrane and the positively charged nanoparticle. However, once you have conjugated the nanoparticle with an antibody, you lose the charge. By losing the charge, you have a mediated endocytosis internalization of the immunonanoparticles. We did it, and we did not find any difference between an immunonanoparticle negatively charged or an immunonanoparticle positively charged. Since we know that positive charge may induce some toxic effects, we prefer to work with negatively charged immunonanoparticles.
I remember the question. You might have said that, but the fact that the metastasis escaped from the treatment, is that due to the fact that they changed their phenotype and they don't express the antibody anymore? Have you checked for that?
No, we didn't. I agree with you, but when it comes to metastasis, this is a quite different problem. And we had some data that are encouraging, but not enough. In fact, we didn't find metastasis in the lung with the immunonanoparticles. We found them, and not in the heart. We found them in the liver, in the spleen, but not in the lung. We do not know why. The reason why we didn't, I do not know exactly why. I have no reason for the time being, unless you will accept two things for the time being. It is one, that metastases know how to deal with antibodies. We are more and more treatments and data coming that metastases are becoming resistant to antibodies. And we do not know exactly the reason, but one of the reasons can be a secretion of acidic pH by the metastasis that prevents the recognition by the antibody. The second one can be that the antigen on the metastasis can be labile, and it can change and escape from the antibody. This is why making it with two antibodies can be of some advantage, but we do not know for the time being.
Thank you.
Next question, please. Okay, thank you. Thank you very much. I'd like to take a couple moments to wrap up for the day. We've had, I think, a fantastic start to the conference. During the course of the day, we've been able to first review things from an emphasis on the telemedical side in terms of body area networks and wireless networks to connect different devices, which may presently be ex vivo, but ultimately end up being in vivo. And then we moved on to look at pathways for nanomedical development and considered a number of very exciting pathways based on the work of the different presenters here today. Pathways that would need to be developed in terms of energy generation capability, the very creative concept and I think clear concept that we need to work downward from the thermal, from the energy that can be achievable from the body's environment, something perhaps on the order of, I think it was 100 microwatts, something in that domain. And then pathways based on different types of substrate. Most of our emphasis was on carbon nanotube substrate, but we also heard quite a few references to other sorts of substrate. And then finally in the afternoon sessions, we began to look at some very exciting particularized applications of nanomedical devices and nanomedical drug delivery capabilities and nanomedical enhanced radiotherapy and radiodiagnostic tools, culminating with Dr. Benita's very excellent presentation about his experience with paclitaxel. So I think we've really had almost a 360-degree perspective of telemedical and nanomedical technology. But perhaps what made me most happy today was the sense that I have received from several of you outside and a couple people from the podium that today there was sort of a realization that in the long run, nanomedical technology implies telemedical technology because both of them are aiming to get us greater and greater spatiotemporal precision, be it in diagnostics or in therapeutics, and that ultimately to provide that greater temporal spatial control of nanobots, we need some type of wireless network capability, and in turn to be able to use wireless networking to its greatest effect, we need some type of nanomedical structure. So there is a real marriage between the two fields of telemedicine and nanomedicine, which is, of course, precisely the purpose of our conference. I want to thank everybody for their kind attention and thank Temple, Genesis, and the staff of Manoir de Sab, as well as our video contractor for providing really superb support to all of us today. We now have a break until the dinner starts in accordance with the program in front of you. So please feel free to relax, refresh, and I look forward to continuing our discussions over dinner this evening at the restaurant downstairs. Thank you very much.